Hello everyone, welcome to the session on Crimean Congo hemorrhagic fever. In this video, we will uncover the intricate details of the Crimean Congo hemorrhagic fever virus, its origins, transmission, its impact on humans, and the preventive measures available to combat its spread. Crimean Congo hemorrhagic fever virus is a tick-borne pathogen belonging to the orthoneurovirus genus in the Neuroviridae family of the Bunia virilis order. The disease was first discovered in 1944 when Soviet soldiers became ill with a hemorrhagic fever after occupying Crimea. In 1969, a virus with identical clinical manifestations was discovered in Congo Basin and was determined to be antigenically identical to that of the virus discovered in Crimea. The linkage of two place names resulted in the current name for the disease and the virus. Now, let us look into the global distribution of the Crimean Congo hemorrhagic fever. The Crimean Congo hemorrhagic fever virus has been reported as a high priority pathogen by the World Health Organization, yet it is unknown to the general public. It has a high case fatality ratio of 10 to 40%. It potentially results in hospital and health facility outbreaks and is difficult to prevent and treat. Where is it circulating? Crimean Congo hemorrhagic fever is endemic in all of Africa, the Balkans, the Middle East, and Asia. This map represents the geographic distribution of Crimean Congo hemorrhagic fever. The countries in red report more than 50 human cases annually to WHO. Those in orange report fewer than 50 cases. Countries in yellow have not reported human cases, but the Crimean Congo hemorrhagic fever virus has been isolated or its presence has been inferred from serological studies, and a transmission competent tick is also present. The widespread distribution of hard-bodied ticks that carry the Crimean Congo hemorrhagic fever virus and its persistent circulation means the risk of spillover from animals to humans is high. In Iraq, the Crimean Congo hemorrhagic fever cases surged from 33 in 2021 to 511 in August 2023, signaling a rapid rise. This graph shows the number of Crimean Congo hemorrhagic fever confirmed cases in 2023 compared to 2022. The blue arrows refer to Iftar Eid week 17 in 2022 and week 15 in 2023, where there was a surge in number of cases. Orange arrows refer to the Adha Eid week 26 in 2022 and week 24 in 2023. There was an increase during the festivals. This could be potentially due to the transport of the Crimean Congo hemorrhagic fever infected livestock from rural to urban areas for slaughter by potentially untrained individuals. This study indicates that there is an urgent need for tailored interventions such as development of targeted public health actions, and also there is a need for international collaboration. The Economic Times reported this virus to be a deadly one because it kills nearly every second patient it infects. It also reported that this could be a cause for the next possible pandemic if left unchecked. Factors such as climate change and the movement of livestock or wild animals may further increase its geographic spread, with the virus recently expanding its reach from the Balkan region to Central and Western Europe. Now, let us look into the virus structure. The virus is spherical in shape with a diameter of 80 to 100 nanometer. The lipid envelope is 5 to 7 nanometer thick and the glycoprotein spikes are 8 to 10 nanometer in length. These viruses are referred to as segmented negative strand RNA viruses by the virtue of their multiple genome segments. The three segments form a complex with nucleocapsid proteins to become a ribonucleocapsid. The virion consists of three main structural proteins: a nucleocapsid protein, glycoproteins GN and GC, which are responsible for recognition of receptor sites on susceptible cells, and a large polypeptide protein, which is a virion associated RNA dependent RNA polymerase with a size of 200 kilodalton. Now, let us look into the molecular biology of the Crimean Congo hemorrhagic fever virus. The genome of the virus consists of single-stranded RNA with negative polarity divided into three segments: the small segment S, medium segment M, and the large segment L. The small segment S encodes the viral nucleoprotein NP along with the small non-structural protein NSS in an opposite sense reading frame. In addition to interactions with the viral RNA to form ribonucleoprotein complexes, nucleoprotein NP possesses endonuclease activity, interacts with host heat shock proteins during intracellular virus replication, and promotes translation of viral mRNAs. The non-structural protein NSS along with NP may have a role in modulating host cell apoptosis. The medium segment M encodes the viral glycoprotein precursor GPC. Compared with the M segment of other Bunya virilis, the Crimean Congo hemorrhagic fever virus M segment is complex. The GPC is proteolytically processed to produce the individual viral glycoproteins GN and GC. A GP160/85 protein that is further proteolytically processed to a heavily glycosylated mucin-like domain MLD and a GP38 protein and a medium non-structural protein that promotes glycoprotein processing and virion assembly is encoded by the M segment. L segment. Consisting of more than 12,000 nucleotides, the large segments of the Crimean Congo hemorrhagic fever virus and related orthoneuroviruses are unusually large for members of the Bunya virilis order. The encoded L protein contains the viral dependent RNA polymerase and cap snatching functions. Interestingly, at the very amino terminus, the orthoneurovirus L protein contains an ovarian tumor-like protease domain. Now, let us look into the life cycle of the Crimean Congo hemorrhagic fever virus. On attachment, the Crimean Congo hemorrhagic fever virus undergoes clathrin-dependent and pH-dependent entry into the host cytoplasm. After entry into the cytoplasm, the viral genomes are converted into positive sense mRNA by the RNA-dependent RNA polymerase, which initiates translation of viral proteins. These proteins also coordinate and produce new negative sense viral genomes, referred to here as cRNA or complementary RNA. These negative sense viral genomes are coated with nucleoproteins and a bound L protein to initiate replication or infection of the next cell. The glycoprotein C is translated into the endoplasmic reticulum, and during trafficking through the endoplasmic reticulum and the Golgi apparatus is proteolytically processed to produce methylglycoproteins along with accessory proteins like MLD, that is the mucin-like domain protein, NSM, the non-structural medium protein, and the glycoprotein 38. Newly produced genomes are packaged into enveloped particles and the virus buds into the Golgi apparatus for release via the secretory pathway. New virions are then released to infect additional cells. Now, let us look into the genetic diversity of Crimean Congo hemorrhagic fever virus. Corresponding with its wide geographic distribution, Crimean Congo hemorrhagic fever virus is a genetically diverse virus, although the nucleoprotein and L proteins of Crimean Congo hemorrhagic fever virus strains are conserved with approximately 95% or more amino acids conserved between strains. The Crimean Congo hemorrhagic fever virus glycoprotein C (GPC) is much less conserved, with divergent strains exhibiting less than 75% amino acid conservation. The genetic diversity of Crimean Congo hemorrhagic fever virus correlates strongly with geography, and clades of Crimean Congo hemorrhagic fever virus segregate based on geographic location. The tremendous genetic variability of Crimean Congo hemorrhagic fever virus can be attributed to virus mutations, genomic segment rearrangement, reassortment, or recombination. Phylogenetic analysis of the S segment of the Crimean Congo hemorrhagic fever virus suggests the strain cluster into 7 major clades. Clade 1, Africa 2. Clade 2, Africa 1. Clade 3, Europe 2. Clade 4, Africa 3. Clade 5, Europe 1. Clade 6, Asia 1. And Clade 7, Asia 2. Interestingly, the Crimean Congo hemorrhagic fever virus strains isolated from similar regions decades apart show strong sequence conservation, suggesting that temporal evolution of Crimean Congo hemorrhagic fever virus within geographical regions is limited. Instead, genetic diversity may arise from long-range migration. Strains of Crimean Congo hemorrhagic fever virus circulating in southwestern Europe cluster with Africa rather than Eastern European isolates, indicating that the long-range introduction of Crimean Congo hemorrhagic fever virus to Europe from Africa, potentially through migratory birds carrying Crimean Congo hemorrhagic fever virus infected ticks. Transmission of the Crimean Congo hemorrhagic fever virus. Exoded heart ticks, especially Those of the genus Hyaloma are both a reservoir and a vector for the Crimean Congo hemorrhagic fever virus. Apart from this, other members of the ixoid ticks such as Rhipicephalus, Boophilus, Dermacenter, and Ixodus may also transmit the virus. Changes in climatic conditions have been suggested to be one of the factors that has facilitated reproduction of the tick population and consequently the increased incidence of tick-borne infectious diseases. Numerous wild and domestic animals can carry the virus, including cattle, goats, sheep, hares, ostriches, and even rhinoceroses. Most human cases have occurred in people involved in the livestock industry, such as agricultural or slaughterhouse workers and vets. However, once infected, people can transmit it to each other through contact with blood or bodily fluids or improper sterilization of medical equipment. Crimean Congo hemorrhagic virus ecology. Ixoid ticks are both reservoir and vector for the Crimean Congo hemorrhagic fever virus. They maintain a two-host life cycle, initially molting from larva to nymphs on small animals such as birds and rodents, then transitioning to larger vertebrates including humans. Enzootic life cycle refers to a transmission stage or cycle during which a disease occurs continuously among non-human animals of a particular region or locality. The virus during the cycle is maintained both transovarially and transstadially. Transovarial transmission is the transmission of an infectious agent from parent to offspring via infection of the developing egg, which subsequently results in infectious adult arthropods. Transstadial transmission occurs when there is transmission of parasites throughout the development of tick life stages, from the engorgement through molting into the next unfed stage of the same individual tick. Epizootic epidemic cycle. Epizootic cycle is a cycle during which the virus is transmitted between non-wild or domestic animals and the primary or accessory insect vectors. Epidemic occurs when infection spreads from one person to another. Crimean Congo hemorrhagic fever cases occur more during warmer parts of the year, mostly the spring and summer. There are no cases during winter. Humans become infected through tick bites and direct contact with infected animal blood or tissue. Transmission can occur while slaughtering infected animals, during veterinary procedures, and in hospital settings where proper protective equipment and appropriate disinfection procedures are lacking. The effects of increasing human movements, vertebrate host migration, and climate change, which affects migratory habits of small animals, threatens to increase Crimean Congo hemorrhagic fever virus infections within the populations. Clinical progression of Crimean Congo hemorrhagic fever. The clinical progression of Crimean Congo hemorrhagic fever presents with four distinct stages. Initial Crimean Congo hemorrhagic fever virus infection is often unrecognized following exposure to ticks or animal husbandry, but transmission may also occur via nosocomial, that is, hospital acquired infection, or intrafamily transmission during the care of sick patients. After infection, the incubation period may be as short as 1 to 3 days, depending on the route of exposure. After the incubation period, infected humans may progress to a pre-hemorrhagic stage characterized by non-specific symptoms such as fever, malaise, general feeling of discomfort, myalgia, which refers to muscle aches and pain, and nausea, which is a stomach distress with distaste for food and urge to vomit. This pre-hemorrhagic phase of disease can then rapidly progress to the hemorrhagic disease in the first week after infection. This is characterized by changes in blood hematology and blood chemistry disturbances. Petechiae, meaning pinpoint round spots that form around the skin, and ecchymosis, which is blue or purple skin discoloration that occurs as a result of rupture of blood vessels under the skin. Epistaxis, which is bleeding of the nose. Melena, that is passage of black tarry stools. Hematemesis, vomiting of blood, and hematuria, that is blood in urine. This phase of disease is characterized by uncontrolled bleeding, liver damage, inflammatory immune responses, and in severe cases, disseminated intravascular coagulation, shock, and death. In patients who survive, recovery begins 10 to 14 days after infection and is associated with return to normal blood chemistry and hematology and development of anti-Crimean Congo hemorrhagic fever virus immunity. Long-term sequelae following Crimean Congo hemorrhagic fever virus is poorly studied. This photograph shows severely ill patients who develop striking pattern of large ecchymosis that are not seen in other types of hemorrhagic fever. This type of ecchymosis is characteristic of the Crimean Congo hemorrhagic fever. Diagnosis of the Crimean Congo hemorrhagic fever. The basic marker in the diagnosis of Crimean Congo hemorrhagic fever is the decrease in the level of platelets and leukocytes. This graph represents the dynamics of blood cells during different phases of Crimean Congo hemorrhagic fever in patients. As we can see, the level of platelets and leukocytes have reduced during the due course of fever. However, the enzyme transaminase tends to increase during the progression of the fever. Enzymes such as aspartate aminotransferase, alanine aminotransferase, creatine and phosphokinase, and lactose dehydrogenase tend to increase. Fibrinogen is reduced, which tends to make a meshwork to bind platelets and proteins to make a clot. An increase in the degradation products of fibrin is observed. Within 5 to 9 days, surviving patients' lab results tend to become normal. Tests on patient samples present an extreme biohazard risk and should be only conducted under proper biological containment conditions. However, if the samples have been inactivated with virusides, gamma rays, formaldehyde, heat, etc., they can be manipulated in a basic biosafety environment. The virus infection can be diagnosed by several different laboratory tests. Enzyme-linked immunosorbent assay is an immunological assay used to measure antigens of the virus or antibodies against the virus. The serum neutralization assay is a serological test to detect the presence and magnitude of functional systemic antibodies that prevent infection of a virus. Reverse transcriptase polymerase chain reaction is a molecular assay which is used to confirm the presence of the virus. Viruses can be isolated in cell lines and the characteristic cytopathic effect indicates the presence of the virus. Treatment for Crimean Congo hemorrhagic fever is primarily supportive. Care should include careful attention to fluid balance and correction of electrolyte abnormalities, oxygenation and hemodynamic support, and appropriate treatment of secondary infections. The virus is sensitive in vitro to the antiviral drug ribavirin. Ribavirin is considered safe with limited side effects. How could we lower the risk of it becoming a pandemic? Prevention of Crimean Congo hemorrhagic fever. Prevention of Crimean Congo hemorrhagic fever requires a multifactorial approach addressing public and veterinary health. Farm workers should wear long sleeves and pants to limit tick bites. Transmission of Crimean Congo hemorrhagic fever in healthcare and abattoir settings. Abattoir refers to slaughterhouses. Can be limited by wearing proper personal protective equipments. Standard barrier personal protective equipment such as laboratory gown, gloves, face shield, and mask can limit the exposure of healthcare personnel to contaminated body fluids. Abattoir workers can be similarly protected by wearing a PPE such as gowns, gloves, and receiving proper training on how to butcher animals. Control of tick vector is also important and may include deployment of acaricides to control tick populations on the farm and livestock. Vaccines are also critical to prevent disease on Crimean Congo hemorrhagic fever virus infection and could be deployed in livestock populations to interrupt the Crimean Congo hemorrhagic fever virus life cycle. Public health directed educational efforts can help to inform at-risk populations to reduce activities that expose them to Crimean Congo hemorrhagic fever virus infected ticks or livestock to recognize the risk of tick bites in transmission of Crimean Congo hemorrhagic fever virus and to promptly recognize the early symptoms of Crimean Congo hemorrhagic fever virus. Inspection and quarantine of animals moving from Crimean Congo hemorrhagic fever virus endemic areas can reduce exposure of humans to Crimean Congo hemorrhagic fever virus infected ticks and prevent the introduction of Crimean Congo hemorrhagic fever virus to new areas. Multiple vaccine platforms for Crimean Congo hemorrhagic fever virus have reached various stages of preclinical testing and include nucleic acid based vaccines such as plasmid DNA and mRNA, inactivated virus preparations, live attenuated vaccines such as recombinant vesicular stomatitis virus, virally vectored vaccines such as adenovirus vectored vaccines, and virus-like particles. There are also subunit vaccines with purified viral proteins. The viral antigen expressed by these vaccines are mostly nucleoproteins and or the glycoprotein precursor. The nucleoproteins are highly conserved and are highly expressed. However, They do not induce neutralizing antibodies because they are not exposed on the cellular virion surface. The glycoprotein precursor C, however, is a target of the neutralizing antibody and it is exposed on the virion surface. However, it is variable in nature. The vaccine-mediated protection may require effector functions such as opsonization, complement activation, or antibody-dependent cellular cytotoxicity by natural killer cells. However, the role of cellular immunity such as cytotoxic T-cell mediated cytotoxicity is not fully understood. It also remains unclear how antibodies protect against the virus. Therefore, commercial vaccines have not yet been developed. Now, a brief summary about the Crimean Congo hemorrhagic fever. Crimean Congo hemorrhagic fever is brought about by a tick-borne virus. It is characterized as one of the 9 priority diseases by the World Health Organization. It was detected among soldiers in Crimea in 1944 and was found to be responsible for an ailment in human in the Congo in 1969. It is transmitted by tick bites or contact with infected animal blood. Human to human transmission occurs through blood secretions, organs, or bodily fluids of infected person. Symptoms include sudden fever, muscle ache, dizziness, headache, sore eyes, sensitivity to light, nausea, vomiting, diarrhea, abdominal pain, and sore throat. Treatment: antiviral drug ribavirin is used with apparent benefits. Controlling infection: standard infection control precautions should be implemented. Reducing the risk: we can reduce the risk of the virus by wearing long sleeves or long trousers while entering tick-infested areas, by using approved repellents on the skin and clothing, by wearing gloves and other protective clothing while handling animals, by avoiding close physical contact with Crimean Congo hemorrhagic fever infected people, by washing hands regularly. These are a few references and journal papers for further reading. Now, let us look into some multiple choice questions. What is the primary vector responsible for transmitting Crimean Congo hemorrhagic fever? A. Aedes mosquitoes. B. Ixodes ticks. C. Anopheles mosquitoes. D. Sandflies. The correct answer is B. Ixodes ticks. Which of the following animals is considered a reservoir for the Crimean Congo hemorrhagic fever virus? A. Pigs. B. Cows. C. Rodents. D. Ticks. The correct answer is D. Ticks. Which family does the Crimean Congo hemorrhagic fever virus belong to? A. Flaviviridae. B. Nairoviridae. C. Togaviridae. D. Herpesviridae. The correct answer is B. Nairoviridae. How is the Crimean Congo hemorrhagic fever primarily transmitted to humans? A. Inhalation of respiratory droplets. B. Consumption of contaminated food. C. Direct contact with infected blood or tissues. D. Sexual transmission. The correct answer is C. Direct contact with infected blood or tissues. Which of the following regions is Most at risk for Crimean Congo hemorrhagic fever outbreaks. A. Arctic regions. B. Sub-Saharan Africa and parts of Asia. C. Western Europe. D. North America. The correct answer is B. Sub-Saharan Africa and parts of Asia. What preventive measures are recommended for individuals at risk of Crimean Congo hemorrhagic fever? A. Use of mosquito nets. B. Tick avoidance and control measures. C. Safe handling of animal blood and tissues. D. All of the above. The correct answer is D. All of the above. Protect yourself from ticks. Thank you for your kind attention.